with fungi. Tomorrow in lab, you're going to have a fun lab, I think. You're going to be looking at fungi. You're going to be setting up edible fungi growing kits. You're going to be doing essential oil extractions as well, which aren't fungi, but that's still cool too. So we're going to be doing a variety of things tomorrow that I think will be very fun. And so you'll learn tomorrow in lab, they'll talk to you a little bit more about fungi, quite different. They'll focus more on the edible and a little bit more on the biology of these things. But I wanted to jump into how fungi can be symbiotic with plants. And so I'm not really going to get into fungal life cycles in any detail, and you really won't see much of that in lab either. You'll get the general gist of it, but from my perspective, when I'm teaching a plant class, I want to focus more on how plants and the associated fungi interact and what can these fungi do for the plants. So I try to be more plant-centric and introduce the fungi and the bacteria around that. Mycorrhizal fungi are incredibly important for plants, and there's still so much research going on. So what you're seeing here is a little seedling, a little conifer seedling. And then if you can see, I don't know if you can see the difference. Well, I think it shows up pretty well where you can see kind of the bigger yellow areas, that those are the roots. Okay, that's the plant roots. But then all of this more delicate looking white stuff, that's all fungal hyphae. All right. And so this is a good image to get across the point that these fungi, these mycorrhizal fungi, are hanging out with the roots. And so they're closely associated with the roots, but they're also sending off hyphae into the soil to do their job for the plant. And so when they started examining this interaction and then doing some careful experiments, time and time again, they could demonstrate that the presence of these mycorrhizal fungi with the plants really improves seedling growth. Even with germination, getting seeds to germinate can be beneficial, but seedling growth for sure. And then just overall growth through its life cycle, the presence of these fungi can be very, very important. So then they realize there's different types of fungi. I'm not going to get into all of them, but this is some of them that we see out in nature. And there are some plants that do seem to not form this relationship. So that's the last line on this table. So the Brassicaceae family is famous for that. But they usually quote something like 85%. I've seen 90% of plant species have mycorrhizal relationships. So here you can see that there's different types. Arbuscular mycorrhiza is the top line. And then ectomycorrhiza, there's orchid mycorrhiza, ericoid, and then there's some others too that didn't make it onto this particular list. So I'm going to be focusing on the top two lines and thinking a little bit more about those. I'd like to start with ectos. So ectomycorrhizal fungi, it's a mouthful. Ecto refers to the surface. All right. And then myco, anytime you see M-Y-C-O-R or just myco, I guess the myco, M-Y-C-O, that is referring to something fungus-like or an actual fungus. And then R-R-H-I-Z-A-L, the end there, rhizal, that's referring to root. All right, so these are fungi that hang out on the surface of plant roots for the most part, but I will explain a little bit more about that. So we see them mainly forming relationships with temperate trees and shrubs. So when you're out hiking around here, most of the trees and shrubs that you will be passing by definitely have these relationships going on. It's just that you don't see them because the roots are underground. So we're thinking about both flowering plants, the angiosperms and the gymnosperms, okay? And some tropical trees also. Now, it's important to realize that you could have certain plants, they can form a relationship with many different mycorrhizal fungi. It's not like a one-to-one strict relationship for the most part. So we see that if you examine the roots of certain trees, for example, you will see several different types of mycorrhizal fungi on them, and that seems to be the norm. And when I was in grad school up at Michigan Tech, one of my fellow friends, grad students, he was up in the forestry department and he was studying mycorrhizal fungi succession. So the forest systems that we were studying, he could show that it took him 5 years to do this study where he was showing the progression of mycorrhizal fungi over time with these different tree species, and looking at just like if you've taken BI-210 and you go out into a forest and they talk about tree succession, plant succession, similar idea underground where you have mycorrhizal fungi that kind of come in initially established, then they kind of fade away as another one comes in and takes over and starts to dominate. And you kind of see that flow of different fungi coming until you see mature interaction developing. So ectomycorrhizal, I studied these when I was for my PhD. That was part of my thesis research, and I just love the idea of fungi that are capable of forming a positive interaction with plants, because really when you look at plant diseases, fungi win. There's more fungi out there that attack plants than bacteria and viruses, and so I learned a lot about that, but it was kind of like there's got to be some good ones out there too. And then I started learning about mycorrhizal fungi actually when I took my first plant phys class undergrad, and I just fell in love with this idea that we have this very intimate relationship between two very different organisms, and yet they team up and they partner up and do good things for each other. And now the more we learn about them, the more we realize there's so much more going on that we didn't realize at first. So here you're seeing some visual images of ectomycorrhizal fungi. On the top left is the lighter colors. Those are the fungal hyphae. So when we talk about how fungi grow, these types of fungi, they form those long strands or filaments. They often say they look hair-like strands. That's a hypha. And then if you were to pluralize that, you'd say hyphae. So if you add an E on the end. So what we see here is lots and lots of hyphae, all compacted very tightly together. So when you eat mushrooms, that's what you're eating. You're eating a bunch of hyphae that have been compacted together to form a tissue-like organization. And then they cover the surface of the root. And here you can see, I'm pretty sure this is probably forming a germus, but ectomycorrhizal fungi, they tend to, when they start to grow on the root, they form that Y shape. You'll see these little tiny Y-shaped kind of fatter looking roots. So if you ever see an overturned tree that blew over or something, look for that Y shape. Find that, you know that you're looking at mycorrhizal fungi hanging out on the root. It's very characteristic, and you can see it with the naked eye. And then this lower left image I like because you have to appreciate that, yes, they form this really tight growth on the surface that's very tissue-like, very flattened, but then they also extend out hyphae into the soil. And so that's really a huge part of their job, is to get their hyphae out there and exploring and looking for nutrients. So that top right image, where you see that white hyphae in the soil system, just trying to impress on you that you have to imagine these hyphae coming off the root and going and foraging. And then this lower right image is an example of a root system that's been carefully cleaned off. And this is like how you would see it with the naked eye. So the top left is greatly magnified, and the lower left is kind of what you would see if you went up to a tree that's been overturned and looked at its roots. You would see that it's usually the lateral roots, the finer roots that are actively absorbing water and nutrients. They tend to be covered with the fungi. So ectomycorrhizal means that they form that very thick growth. Excuse me. So we refer to it when it's growing thickly like that on the surface of the root. We call that a mantle. I would like you to know that. So know the hyphal term, hyphae, mantle, and so if someone was looking at that picture, they'd say, "Oh, look at that fungal mantle on the root surface. All right, that's how they would express it. And so it's very characteristic of ectomycorrhizal fungi. And anytime you see like that the word down on the lower left, ectomycorrhizae, anytime you see anything like that, what they're referring to is they're taking into the plant root and the fungus together. Then they start saying, oh, look at the ectomycorrhizae. That's just the symbiotic. So these are different species of fungi that are forming on these roots, and you can see that they look different, visually. They're different colors, slightly different sizes, different morphology. And so these fungi are not all identical. There's a lot of different fungi that are classified as ectomycorrhizal fungi, and especially around here. And so if you were to take one of those roots and cut it into a cross section, you might see something like what they've drawn there on the left, where you see that really thick growth of fungal hyphae on the surface. So that's your mantle along the around the perimeter there that you're looking at. So all of this is the mantle, okay? And then you see the root cells below that with the vascular tissue in the center. And the way the artist drew this, you can see that there's hyphae that have grown into the root tissue. Okay, so all of that kind of tannish stuff that's showing up in between the cells in that net-like growth pattern, that's fungal hyphae. So not only do these ectomycorrhizal fungi grow very thickly on the surface, they also send some hyphae into the root tissue, but it's all in between the cells. They just kind of go in between all the root cells. And if you can imagine, if you look at that, it does look like a fishing net. And so they often refer to that formation like that in between the cell. The person who came up or first discovered this, his name was Hartig, Dr. Hartig. So they call that growth morphology a Hartig net. So that Hartig net just means have the fungal hyphae growing into the root, but it's in between the roots, or the root cells, like in the cell wall space. All right, so it's all cell wall. The right image is trying to show that you get a shift in the root morphology when you get this interaction going between the plant and the fungus. And so normally roots they're just straight. That's usually how roots grow. They just kind of grow straight. Once you get this interaction going on between the plant and the fungus, it tends to stimulate the finer roots to grow into that Y shape. And then the fungus tissue, the hyphae, cover it very thickly like a tissue. And so again, that's what I mean by that. If you see that within root system, you know that you're looking at mycorrhizal fungi established there. And so this idea of this interaction, when they realized that this was happening, and then they started, well, what kind of plants do this? What kinds of fungi? And they realized how common it was. Then they started doing lots and lots of controlled studies and comparing, what happens if you do have the presence of the mycorrhizal fungi or if you don't, looking at plant health overall. And so this is a really nice image showing on the left plant roots that were treated with mycorrhizal fungi compared to the same species of plant on the right that was the control without any mycorrhizal fungi. And you can see a huge difference visually, very obvious difference. And so you can see this with the naked eye. Like I said, as you're out hiking around, you can tell the difference between roots that are colonized and those that aren't. Once you train your eye a little bit, it's not terribly difficult. So here's some beautiful images of different plants, roots, and their mycorrhizal fungi, the ectomycorrhizal fungi. So of course they wanted to know why. Why would this partnership develop? What's the payoff? If for so many plants to do this, it must be beneficial for the plants. It's not like just one little species of plants does this. It's, the vast majority of plants that have been studied. And so here they're looking at the Hartig net more carefully under the microscope. And so all of that dark kind of greenish growth And so if you can imagine the fungal hyphae hanging out that close to the root cell, it's really a good strategy for doing some exchange of nutrients, water, chemicals in general. Okay, so it's a nice way to easily get some flow back and forth. And so they started looking at this more carefully, and they did realize that yes, the presence of the mycorrhizal fungi does help the plant get access to key nutrients from the soil, and it also allows for other chemical communication that's going on between the fungus and the plant. So they started and water too. Of course, water uptake, the fungal hyphae is really good at scrounging up water in the soil and getting that to the plant. So water uptake has greatly improved in the presence of fungi. This is a nice microscope image because it's showing that very thick mantle. All the fungal hyphae pressed very closely together, growing on the surface. All that blue that you see on the surface. And then the Arctic net, you can see again the blue is the same that picks up on the Arctic net. So very, very nice specimen, slide specimen. So if you can imagine, if you're the plant and you have your roots covered like that, that can be a good thing too, because those are your finer roots that are in the soil, typically that are well colonized by the fungi. And your finer roots tend to be kind of a food source for other creepy crawlies in the soil. And so you are getting some physical protection too, if you do the relationship with ectomycorrhizal fungi. And so they can see less herbivory damage on those finer roots like this. So I was really interested in ectomycorrhizal fungi. We were doing a big research with different universities. Michigan Tech was one. Then there's a couple downstate, I think one in northern Wisconsin somewhere. We were looking at these different forested systems, and my mentor won a grant to look at the health of these forested systems. This is back when the Navy was going to put antenna systems in the Upper Peninsula to communicate with submarines in the Atlantic Ocean. Doesn't that sound kind of weird? It's like we're not even close to the Atlantic Ocean. What the heck? But they did get the grant. They did put these antennas. So they had above ground antenna systems. They had below ground antenna systems. It was a big deal. If any of you are from the UP, you might want to chat with your mom or dad or grandma about this, because I mean, there's like people protesting this and chaining themselves to the fences where the antennas were going to get installed. It was a big deal. They ended up getting installed, and so my mentor got a grant with these other universities to ask the question, will these antenna systems do anything to harm the local environment? So there was some concern about that. And so part of our project was to examine the health of the forested systems. And it's kind of hard to do that when you look at tree species because they grow so slowly. Sometimes you don't see the stress for many years beyond the actual stress. And so our part of the project, a small piece, but our part was to ask what happens to the microbes in the soil system with these antenna systems. Because that you can measure very quickly, you can see shifts very quickly happening with a disturbance. So we were looking at mycorrhizal fungi. I was also looking at some beneficial bacteria. And then I kind of went on an offshoot and I was looking at some other ideas related to this plant microbe interaction. But anyways, the upshot, by the way, at the very end of the study, at least our data set could not find any statistically significant differences. Presence or absence of the antenna did not seem to alter the microbial ecology of the ecosystem. So it was kind of funny how all that kind of nuance. So when you're out hiking and you're walking by some trees, you see some fruiting bodies or mushrooms, chances are in this area, you're looking at ectomycorrhizal fungi that have sent up some mushrooms. And you're going to learn more about mushrooms tomorrow in class, but basically anytime you see a mushroom or fruiting body, basically what's happening is that's the the way that the fungi is going through sexual reproduction. So they're basically having sex and shooting up these fruiting bodies in order to accomplish the completion of their sexual reproduction. And so that's why you often see fruiting bodies kind of hanging out right by the base of the stem of a tree, or just out from it a little bit, and often sometimes in a circle pattern. They can be shooting up through the soil. So they are pretty easy to spot if you've been out hiking. Okay, questions on ectos? Ectos, the reason I chose to pick ectomycorrhizae to study is that they're easy to work with in lab, relatively easy. They grow slowly, so you have to have patience, but you can grow them separate from their plant partners and they'll survive. And so they're easy to grow on an agar plate and study. And I was doing some gene expression studies looking at what happens when they start to interact with the associated plant host. This tracks a little bit. The others we're going to mention are the endomycorrhizal fungi. And so these endo, within, what they're trying to say here with that is that we have to think a little bit more about what's happening in the root tissue, because we see some interesting things going. So endomycorrhizal fungi, that's one way to talk about them. They're also called VAM fungi. You might hear someone say VAM fungi, which stands for vesicular mycorrhizal fungi, which is a mouthful, so that's why we shorten it to VAM. Now I'm seeing more and more published studies where they just drop the V, the vesicular, and they just call them AM fungi. It's a little hard to say, but I see that. So vesicular or muscular, the reason they say that is that the fungal hyphae, when it's in the root tissue, they can actually balloon, some of the hyphae balloons up like a balloon, a vesicle. And it has that roundish shape, as you see in the lower left, number 1 there. So those are fungal vesicles. They just kind of, it's part of the hyphae that balloons up. And then the fungi can also form these called arbuscules. Arbuscules, if you look up that word, it means dwarf tree. And if you look at that lower right image, that's a great electron microscope image of a dwarf tree. That's what it looks like. So that's an arbuscule. Again, that's just all fungal hyphae that differentiates in this tree-like or shrub-like growth pattern. And they think that the fungi are doing that because it really helps with surface area to volume. And it can really help with nutrient exchange and chemical exchange. It facilitates a little bit better flow of nutrients back and forth and other chemicals. So they realized that there's these other group, they started identifying all these different types of mycorrhizal fungi. With the endomycorrhizal fungi, they are finding they're way more common than the ectos. So about 80% of all vascular plants. So that's we have seedless vascular plants, which we'll get into. We have gymnosperms and angiosperms. So those are all vascular plant groups. So 80% of all 3 of those types. And I've seen that push more to like 85, 90, depending on who the expert is that you're talking to. So a lot, the vast majority. So lots of herbaceous plants that you see that are just, grow in one season and die off. They have these relationships with endomycorrhizal fungi. So your grasses, including your lawn grasses, other herbaceous plants like tomatoes, oranges. So lots of commercial crops that we rely on to feed all these billions of people will have this type of relationship. And then there's some trees too that will form this relationship. And you can have, like, especially tropical trees. Tropical trees tend to have some ectomycorrhizal fungi that they hang out with, but also some endo on their root system. So you can have both. It's not either or. And so unfortunately, there's a lot of literature out there that kind of explains this group a little inappropriately. So I want to make a very careful clarification so that you can kind of be the ambassador and let people know that it's not exactly true what they're saying. So this is a microscope image of root tissue. And the endomycorrhizae are staying red. And you can see some vesicles and what looks like arbuscles. But the way, I mean, when you look at this image, wouldn't you say, oh, I mean, if I said, what do you see here? You'd say, well, I see a bunch of red hyphae in the root cells, right? I mean, that's what it looks like. And I understand why you think that. That's what it looks like. But when you do very careful studies, that you have to realize when they do these kinds of preps, there is some limitation to the visual ability. When they've done electron microscope pictures of what's really going on between the fungal hyphae and the root tissue, it turns out the fungal hyphae aren't in the cells, like floating in the cytoplasm. They're not doing that. But that took very careful technique and an electron microscope to tease that out. So that's why you see so many published things saying endomycorrhizae grow in the root cells. And that's not entirely accurate. So what they do, you have to imagine that the fungal hyphae, so with the ectomycorrhizae, I said they just grew in the cell wall, in between, right? So they're just in cell wall. With endo, what they do is they can grow into the cell wall space, when they get into the root tissue, they're in the cell wall space. They can go through the cell wall, and then they can bump up against the cell membrane of the root cell. But they don't penetrate the cell membrane. They don't do that. They just kind of push up to the cell membrane, and that's it. And so it's hard to draw that though. And so I understand why there's a lot of misunderstanding on this. But since we're in this class, please do not think that the hyphae penetrate through the cell membrane. They just kind of bump up to it and hang out there. And it's still really good because it allows for nice communication and flow of nutrients. So it's still really effective. And then the artist here is drawing the vesicles and arbuscules. And then they're also trying to show how the hyphae comes off the root. So again, with this fungus, a huge part of their job is to get their hyphae out into the soil. So the hyphae with endomycorrhizae, they grow a little bit on the surface like this, but they don't do a mantle like with ectos. So they're very kind of lightly growing on the surface. And then some of those hyphae go off in the soil, and then some of the hyphae goes in to the root tissue. So you have hyphae kind of doing different jobs. Some are destined to grow into the root tissue. Yes. Yeah. So you have to imagine them kind of weaving their way around all the root cells and just hanging out in the cell wall space. And then endos, they can take it a step further and they can go past the cell wall and bump up to the cell membrane. And so it's a more intimate association because they can actually make contact with the cell membrane of the plant cells, the root cells. And so the root cells are right next to the hyphae. The cell membrane is right next to the hyphae. Let's do a eye clicker. Let's see if we can do that. Whoops. I don't know if we'll. Where'd it go? Whoops. Oh, my computer. Just love it. All right, let's do it this way. Oh my goodness. It's frozen. All right, I'm ready to do this. Let's try that again. Okay. So is it letting you in now? All right. What would be a consequence if fungal hyphae did breach the cell membrane and got inside the cell? So it's floating around in the cytoplasm. We all chimed in? And of course, the graph decides not to show. 86% of you chose D, which is correct. So I'm pausing here to ask you this question because this is why I get a little upset when I see in the, in the literature that they're saying, oh yeah, the hyphae get into the cells. It's like, no, because this would happen. You would get this idea that that's a pretty serious breach of physical boundaries, like boundaries, you gotta have boundaries. And so if a plant is actually getting a foreign entity breaching its cell membrane, not only you have to think about fungal hyphae, the cell size or the size of the fungal hyphae and the plant cell. That could kill the cell, first of all, because the fungal hyphae is pretty big. Cell's pretty small, so there's that size issue. Could kill the cell. It probably is going to trigger the plant going, yikes, I'm getting attacked. Something's attacking me. And then they would kick into a defense response. And careful research has shown that to be true. That even though it's a mycorrhizal fungus, if you artificially create, like, the experimenter goes in and creates a damage so the fungal hyphae can get into the cell, the plant does initiate a defense response. And so, yes, they can hug up to the cell membrane. The fungal hyphae can do that and make contact, but it doesn't actually penetrate through and actually exist in the actually in the cytoplasm. Okay, is that clear? That's going to be an exam question. I really want that because there's just so much misinformation out there. So I can make a point another way. Okay, so you've got to respect the boundaries, right? So they've looked at this carefully, whether it's ectomycorrhizal fungi, endomycorrhizal fungi. It's like, okay, if so many plants are forming this relationship, there's got to be a payoff. There's got to be some kind of benefit to the plant and then probably to the fungus too. So it's probably a win-win. And so, of course, they've done thousands of studies by now looking at, what happens when plants go into this relationship versus if they don't. And they can see that you get a huge increase in uptake of certain nutrients. So I've ranked these in order, P and K. So they see, in particular, phosphorus uptake is greatly improved in the presence of these fungi. And then also nitrogen and potassium, but especially phosphorus. And in fact, when I, one of my grad students, Jessica, she was doing her grad research she was looking at red pine seedlings and ectomycorrhizal fungi. And of course, part of the experimental design is she had to get this relationship to establish. So she would, she got the seedlings growing, she spiked in the fungi into the soil, and they wouldn't form a partnership. They wouldn't do it. And we're like, oh, okay, so we had to start all over. And the key thing we had to do is we had to be a little mean to the plant. We had to create a soil system that was really low in phosphorus. As soon as we did that, then the plant went, I can't seem to get enough phosphorus to form a relationship with these fungi. So we kind of persuaded the seedling to go ahead and get into this relationship. And as soon as we did that, we denied it a significant amount of phosphorus. Then her experiment worked out really well. So great uptake in water in the soil system. The fungal hyphae is growing off the roots and getting into the soil, getting access to these minerals, to the water. We see, of course, that that's going to stimulate seedling growth and overall biomass by harvest. It helps protect them. You know, I talked about how with that thick mantle, you might get some physical protection. There's also chemical protection. One of the reasons why I studied the mycorrhizal fungi as a grad student is because they're good biocontrol agents, or can be. So I was looking at that capability of some of the fungi I worked with. And then if you think about it from the fungal perspective, they got to have something to gain from this partnership too. And definitely they do. They get lots of nutrition from the plant, primarily sugar. In photosynthesis, the plant actually gives a lot of its sugar production to the fungus. Some studies have shown that in some of the studies done by Dr. Samard with ectomycorrhizal fungi hanging out in primarily conifer forests, she was showing that roughly 30% of the sugars the plant makes goes to the fungi. So that's a lot of sugar that's going to the fungi. And so the tree is definitely demonstrating that this is a valuable partnership if it's willing to send that much of its resources to its fungal partner. So definitely a win-win. You can play around with these if you garden this summer. You can order mycorrhizal fungi off of Amazon and other places, and they usually come in a mix, and you can apply it into the soil. There's also formulations you can apply hydroponically, and you can see this is from the Great White idea here. I bought mycorrhizal fungi from that company numerous times for some of the projects the students do, and we've had really good success with that. And this is off their website, just trying to show the differences in growth of the plant, present or absent. Questions on the endo mycorrhizal fungi? Well, I could go on and on and on, but I will resist because these fungi are amazing. But there's a few more things I want to say before we get out of the soil. And so here, thinking about this idea that, you know, in a soil system with the typical plant putting its roots into the soil, certainly it's going for water, needs water from the soil, but we also need the inorganic mineral nutrients that the plant needs are in the soil. And so that's a huge job of the root system. Here you're seeing a comparison between what if you feed your plant synthetic fertilizers, inorganic fertilizers, or if you apply some kind of organic amendment, like manure, or there's all kinds of organic. I like to use worm castings. And so here they're trying to show the difference that they're very different strategies. If you use inorganic, you buy the pellets or fertilizer, NPK fertilizer, and mix it into water, and irrigate your soil, yes, the plant will get direct access to that. And so it can be a very quick boost to the plant. On the other hand, with organic fertilizers like manure or something like that, it's a longer process. It takes a little bit of breakdown, and microorganisms are a huge part of that process. And so the microbes in the soil, in a more natural ecosystem, you need those microbes there because, you know, more natural ecosystems aren't getting fed a bunch of synthetic fertilizers. And so if you don't have a soil, in the sense of lots of good microbes in the soil, along with just the soil composition itself, you got to have a good population of microbes in the soil for healthy plants. And so when you look at soil, just at different soil systems, and you know, you probably, many of you either have or will our soils class here, so this is just a tiny taste of what you'll get in that class. But when you look at the soil system, it's composed of inorganic particles, organic particles. Then, of course, don't forget you have the water in there, and then you have gases that are dissolved in the water, just like, whatever gas composition we have above ground, that's what we see below ground. So we have, oxygen and nitrogen and CO2. We also have things like hydrogen gas, methane going on under there too. Just we have trace amounts up here too. And so soil can vary tremendously depending on the inorganic composition. Do you have sand? Do you have silt? Loamy system, clay system? And so those inorganic particles, they vary in their size. That can have a huge impact on the texture of the soil. And it can really have a huge impact on root growth, how well and how easily the roots grow through the soil. And so here in this lower image, you're seeing a root interacting with the soil system, and you see a lot of blue. That's the water they're trying to get across, that there's water dissolved in the soil. And then they're trying to show some different particles in the soil. So it's complicated. Soil is very, very complex, very complicated, lots of differences across different geographic regions. So there are a couple of things you could say, though, that's kind of consistent with these different soil systems around the world. When you start looking at them and looking at the soil chemistry, you want to think of it that way, what they're finding is that the soil system have an overall, the inorganic particle, they have an overall net negative charge on the surface, all right? And so, and I won't get into all the details of why, but that tends to hold true no matter what soil system you're looking at. So if they have negative charges on their surface, that means that you could have then some positively charged molecules easily attached to, basics of chemistry, positive and negative charges attract each other, they'll bind with each other. And so these soil particles will have cations, positively charged ions, on their surface, bound to them. Okay, so some of those cations, a lot of those cations, the plant needs. Those are minerals that the plant needs. But the plant has limited ability to just grab these cations off the soil particle, break that bond and get it. And so they're limited. They can only really get access to these minerals once they've been unbound from the soil particle and they drop into the water solution of the soil and get dissolved. So once the minerals are off the soil particle, dissolved in the water, in the soil, then you can have the plant roots easily take that up. But it's not like the roots have a really easy job. It's kind of challenging for them. There's very few cases where they can successfully grab minerals off, so to speak, off the soil particle. They have to have very special enzymes and mechanisms in play to do that. Not all plants have a full suite of options like that. Okay, so that begs the question, all right, so we have all these balls here on the picture supposed to be kind of the balls of the soil particle. It's there for the taking, except the plant can't easily take it until those minerals come off the soil particle and are in the water. And so now you start, I hope, to see that, well, there has to be some mechanism in play to get that to happen. I just found out roots can't really do it that easily. So there's a couple of strategies in play that can help the plant. One is called the cation exchange process. And again, you take soils and learn a lot more about that. But that is one great way for the minerals to come off the soil particle and get into the water. And so the other big idea is the microbes in the soil. Microbes have a more plentiful skill set in terms of their ability to release particular chemicals and enzymes that can easily cleave off these cations and get them into solution. And so that's why you hear so many farmers saying, you need a healthy soil in terms of good microbial population so that they're busy helping the plant get access to these minerals. And so here, I love this drawing here with trying to show qualities and microbes hanging out in the soil and near the roots. And then little image trying to show how you can find microbes attached to these mineral particles. And so they're right there where the cations are and can help release those cations. And although we've been really focusing in on fungi, don't forget bacteria are very plentiful in the soil. Thank you. So here, the top right image here, you're seeing a clay particle, and they're showing the white, those are the negative charges. And then you have these cations, potassium, calcium, magnesium, hanging out bound to the surface. So the cation exchange is this idea that you can, some of these minerals, these cations, have a real high binding affinity, like they're really well bound to the clay, and others are more loosely bound. And so we're looking at what their binding affinity is. Is it high, medium, low? And what we find is that these minerals can kind of bump each other off, so to speak. And so that's what we mean by cation exchange. One cation can come along, bump another one off, and the one that gets bumped off goes into the water. Hopefully there's plenty of water. And so there's these lyotropic series that you can look up. Here's one. And so what they're showing here is that the way they traditionally draw it out is that the cations on the far left have the highest binding affinity in the row. And then the ones as you go more and more to the right, the binding affinity is lower and lower and lower. So what this is saying is, for example, hydrogen H+, that cation, that one has the ability to replace or bump off calcium, magnesium, potassium, ammonium, sodium off the particle. So if you can imagine, and then in this image, what I'm trying to show is that as when you are looking at an active soil system with plant roots and microbes in there, you do get release of CO2 because they're respiring. So they're giving off CO2, and that creates a situation where H+ or the hydrogen cation, the proton, gets produced. And so you naturally get protons, H+ in the soil just by living organisms. And so that can then replace or bump off the cations with lower binding affinity. And then those that get bumped off go into the water. So this happens to a pretty high dynamic with rain too. When rain is falling and it's percolating through the soil, you have this cation exchange ramping up with the flow of water kind of distributing the protons and just getting all of this cation exchange. So that's a great way to get certain cations off the particle and into solution, and then the plant roots can then take it up. Make sense so far? Okay, we'll finish this idea on soil systems and then get into other symbiotic relationships. We have to talk about how fungi play a role in lichens too. So we'll get into that on Friday.